So I'm Christy, and here's my colleague Valerie, and we're excited to share with you our own experiences with inferring cell-cell communication in spatial transcriptomics data. So why study cell-cell communication? Perhaps all of us come from different backgrounds, but it's no surprise that cell-cell signaling defines many biological processes important for physiology, such as those in driving developmental processes, inflammation, and disease. It's really easy to appreciate how complex an organism is. In this in vivo live imaging of a lymph node, you can really appreciate how each individual cell interacts with many in its environment, which is driven by hundreds of different signals, such as those important for chemotaxis or inflammation and other processes. Cells can receive signals from various distances. Of course, what we're interested in is understanding how local signaling in the immediate tissue neighborhood plays a role in cell states. So we have, of course, juxtacrine signaling, which is contact dependent, and this includes signaling that is mediated by cell to cell as well as cell and matrix. And we also have secreted paracrine signals that diffuse locally within the tissue. And what is currently understood is that the target cells receive the signal if they, one, express the target receptor of the ligand that is being secreted in its local environment, and two, it's located near or adjacent to the sender cells. Of course, one other type of signaling that is not discussed as much in the context of spatial transcriptomics is endocrine signaling from distal organs. And in this situation, there are endocrine cells that secrete hormones through the bloodstream that are able to reach distal sites within the body. But for now, we are focusing on local signaling in tissue of interest. And of course, when we lose the spatial context, it's very difficult to assess relevant signaling that is important driving these cellular states. So current methodologies to study cell-cell communication includes single-cell technologies. Of course, I just mentioned that the main drawback of single-cell technologies is that once you dissociate the tissues and you separate the cells, you lose the proximal information of like how close one cell is to another. This is mitigated by advances in spatially resolved technologies, where there are two major classes of these technologies. One is the in situ capture-based methods, where tissues are sectioned onto a slide with capture probes, which can hybridize to target RNA or target probes. And the benefits of this method is that it can capture nearly the whole transcriptome, but unfortunately at a lower spatial resolution. We have also another class of methods, which are deemed imaging-based methods, where they may have lower gene coverage that is limited by the probe set that you use to target your genes of interest, but they have higher spatial resolution, sometimes even subcellular resolution. And of course, with these spatially resolved technologies, one can infer cell-cell communication through the expression of both the ligand and target receptors in each individual cells, and you still maintain the physical distances between the cells themselves. So in our seminar, we are focusing on how one may infer cell-cell signaling through a popular technique called the Visium assay, Visium or CyAssist. So this is a capture-based assay where within one capture area, there are 50 micron spots, each with a unique spatial barcode. And depending if you're using Visium or CyAssist, at some point you do, in both methods, you do in situ probe ligation to target mRNA in the tissue. And you then release the ligated probe through RNA digestion and tissue permeabilization. And these probes are hybridized to the poly-DT tail of this capture oligo. And from there, you can synthesize your library and sequence and basically recover whole transcriptome profiles at spot resolution of tissue, of your tissue of interest. And so this is what the Visium assay looks like. Here I'm showing an example of one of our tissues that we work with, where we have overlaid a mask of the Visium spot locations. And you can see that within each spot, there are multiple nuclei present. And on the 10x Genomics website, they would claim that it's 10 to 15 cells captured per spot. But in reality, depending on your tissue, the cells may be much more dense. And in our situation, there are some spots captured up to 50 cells, which can lead to certain problems because now each spot is a bulk signature from a mixture of cells. And one of the important things one must do to infer cell-cell communication is to first identify what type of cells are contained within each spot.
So in this case, we perform a procedure that is called deconvolution. And the goal is to kind of make those gene spatial profiles from each individual spot in Visium, make them more interpretable, and see how many cells of each type is presented in each spot. So there are a variety of tools to do that. And what we basically need is our Visium dataset and single-cell RNA-seq reference dataset. So once we annotate our cells in single-cell RNA-seq dataset, we could use, for instance, tool called cell-to location, which we successfully use in order to infer the composition, cell type composition of each spot. And this tool uses Bayesian inference in order to learn signature of each of this gene expression signature of each of the reference cell types, and then to fit the model in order to explain the gene expression from the Visium data and ultimately given as an output numbers of each of the cell type per spot. So in this case, this is the lymph node tissue, and we see, for instance, B cells and follicular dendritic cells localized into germinal centers, as well as myeloid and T cell compartments in those tissue. So we kind of see that some cells probably colocalizing in this data. So we kind of want to know, approach this colocalization question more systematically. So what we can do downstream of this inference of cell type composition is a procedure called factorization of this data. So in this case, we take not gene expression count matrix, but spot abundance, cell type abundance composition matrix. And then we perform procedure that's called factorization of this matrix in order to learn some sort of latent factors, which only represent like linear combinations of abundances of cell types. And in order to ultimately capture some sort of interpretable domains of the tissue that would represent some anatomical structures. Like here, we can see that factor 1 is enriched with T cells, so kind of like T cell zones. Factor 3 is a marginal zone of the germinal centers, and also some other types of relevant tissue structures within lymph node. Now that we address the question of convoluted signal in our Visium data, we could try to infer cell signaling from this data. And there are a variety of tools to do that. Most of them were so far developed for single cell analysis. There are more and more appearing for spatial analysis as well. And they rely on different models and assumptions, but what's common for them is kind of they use co-expression of ligands and receptor mRNAs in respectively sender and receiver cells to kind of model and infer whether those cells communicating. Now, as Christy pointed already, spatial data has some important advantage here because we directly see which cells are proximal, so which are at all can interact, as opposed to single cell where the spatial context is lost. What's important to note that all those cell cell communication tools rely on manually curated databases of ligands and receptors that could interact at all with each other. And they vary substantially, as this heat map demonstrates, in terms of their coverage and at all molecules that are annotated there and included. So it's important to note that depending on the database that you use for inference, you could obtain different results. So one of the tools that we used in our experience is called COMMOD, and it uses something called optimal transfer theory to infer ligand-receptor interactions from gene expression data. So it starts with, it relies on ligand-receptor database, and it knows all the possible combinations of ligand receptors that can interact. And then on our spatial data for each spot, we impose kind of a spatial constraint which tells which are the neighbors for those spots that we consider to be neighbors of the spot. So to account for which interactions are spatially possible at all. And then with this distance constraint, this model tries to distribute ligands molecule counts to the respective receptors in an optimal way. And it uses something called the cost function to optimize this sort of ligand receptor inference. And there are a few important advantages of this model. One of them is that it accounts, for instance, if we have two distinct sources of the ligands in our tissue and a single receptor here, this model will account for the competition between those ligands, between each other for the interaction. And ultimately, what it will show you is spot-spot pairwise signaling strengths as an output, as long as it will calculate the directionality of the signaling on the whole tissue level, which would give us a visual idea of what type of processes potentially going on within the sample. So these are a couple of examples of chemokine signaling, exemplified by CCL90, CCR7, ligand receptor pair, as well as contact galactin with the PTPRC signaling. And without going too deep into lymph node biology, we can just have a glimpse of an idea of what it might mean by comparing this localized signaling with the cell type abundances that we inferred previously in our lymph node, which in the first case, it kind of, most of the cases look like how T cell zone looked like in our previous analysis. And in the second case is how interferon responsive B cells localized. So this is kind of give us some visual idea of what type of process could be going on in tissue. But we probably want to study it more systematically, despite we have convoluted signal from spot levels. And we'll come back to that later in a couple of slides. But probably last thing I wanted to mention is how to rank this signaling. And by ranking, I mean how to understand which one is probably more important, more significant, and to pay attention to in downstream analysis. And in order to do that, one important spatial parameter that could be inferred is called Moran's I. And it's basically some sort of a score that predicts how autocorrelated signal is. So here there are some examples of how starting from anti-correlated to no correlated or very correlated signal would look like. But so to give the visual idea of that this score will tell you how localized the signal is. So meaning that, oh, there are probably particular region in tissue enriched with this localized signaling. So we could rank our inferred ligand receptor interactions from our Visium data, and we can rank them using Moran's I. And here are examples of top autocorrelated spatially signalings, like in those examples, or not correlated spatially. Another tool that we use in our analysis is called Liana, that helps to predict ligand receptor interactions in spatial data. And it's actually not just one tool, it's a few different functionalities that are implemented in this package, such as cosine similarity or Moran's R, a different technique that allows to spatially find spatially correlated co-expressed ligands and receptors from the database. And a nice thing about this tool, to give you a bit of a sense why we like it, is because it's natively implemented with other tools from this ecosystem, such as for gene enrichment analysis or for different multimodal analysis approaches, which we'll touch upon later. So this is the example of ligand receptor inference from Liana Plus, and demonstrates different relevant ligand receptor pairs for lymph node biology.
So of course, now that we're able to assess which sort of ligand receptor signaling is present in our tissue, now we would like to link our spatially confined signaling activity to specific cell types. And what are some ways that we can do this? So in our experience, we tried out a tool called MISTY. It stands for a multi-view intercellular spatial modeling framework, which essentially captures functional relationships between distinct features that is defined by certain spatial constraints. So in MISTY, we would like to understand how inferred cell abundances, which was extracted from a convoluted signature in a spot, can predict the total variance of a target ligand receptor signaling pair. And in MISTY, there are different spatial views one can use. So the default view is the intraview, which is within an observation. So a spot in our case, because we're using Visium or SiteAssist, but maybe for some of your cases, you have single cell resolved spatial data, which is great. Within one cell or one spot, how can one type of feature, such as a cell abundances from a specific cell type, explain the variance of a specific ligand receptor activity? Within this, and this is modeled through a random forest model, but of course there are other types of models one can select in this framework. One can also add additional spatial views, such as what they called a juxtaview or a pair view, to understand whether when you add features from adjacent spots or adjacent cells, either in the neighborhood itself or in a greater spatial tissue context, like can that improve the total predictive score for your target ligand receptor pair? So in our case, we want to demonstrate our own experiences using the intraview because we felt that with the Visium data specifically, each spot already contains multiple cells in a specific neighborhood. And we wanted to understand how the mixture of cell type abundances may explain signaling activity. And some of the outputs of this MISTY analysis includes a rank of features. So it ranks by importance which cell abundances explain the variance of every ligand receptor pair that you have in your data set, and whether additional spatial views, such as these juxtaviews of features in adjacent spots or cells, or larger pair views, can increase the predictive relationship between these features. And so in our case, we wanted to see, can cell type abundance predict ligand receptor activity? And so here, to demonstrate, we used the ligand, we tried to predict ligand activity from the Komoot output, from the cell to location output, which gives us the cell abundances. And we do see biologically relevant signaling, ligand activity specifically, that correspond to the presence of these specific cell types in that same location. So for example, FDCs, follicular dendritic cells, they have been demonstrated to secrete these specific cytokines or chemokines to recruit certain immune cells, and this has been captured in this type of analysis. One can even take it to the next step and ask whether signaling activity can explain the enrichment for certain gene expression modules. So now we want to predict, for example, specific biological Go terms by the activity of ligand receptor, or by ligand receptor activity. And in this case, we used the Liana Plus output because this framework is very flexible. You can honestly use anything that you've analyze in your data set and try to draw these functional relationships between these features and those features. And here you can see, for example, that these different immune processes that were captured at the spot level can be explained by specific activity of these ligand receptor pairs. So we can recommend this framework for trying to integrate all these different information together. So as a recap?
So to recap, we kind of try to explain on how to infer spatial signaling from the Visium data and how to also mitigate the problem of convoluted signal coming from a few cell types per spot. So, and we used cell location to do spot deconvolution, and then ultimately we used MISTI framework to explain the signaling from the perspective of cell type abundances. So this sort of spot level inference is still something we guess about our data. So ideally one would have to validate it with using spatial single cell resolution methods. However, it's worth noting probably that it is still Visium in this case we find helpful as an approach because it's untargeted, so we could get an initial idea on the ligand receptor candidates and the type of interactions that are there. Plus, it's probably the scalability is an important factor if you plan your study, because other techniques might be a bit better in terms of resolution, but a bit less scalable in terms of
costs
or data management, yes. But touching upon that, there are probably some even more exciting opportunities coming with the advent of resolution in spatial methods. And there are a few very promising technologies for this sort of analysis that are reaching or faithfully give you single cell spatial resolution, such as StereoSeq, Visium, HD, or Xenium. And one could probably do even more sophisticated analysis with that, because in this case, you would actually deal with single cells. So as an outlook and perspective we would like to offer, we would like to briefly discuss a few approaches. For instance, there was a recent publication of the tool called FlowSeq, and what their innovation was that they, on top of co-expression of ligand receptor inference, they also included the account for transcription factor that is downstream of this given ligand receptor pair. So they kind of include TF molecule expression into the analysis in order to infer not just potentially how much ligand or receptor molecules were there being expressed, but actually potentially triggered the signaling of the downstream pathway within the cell. And this is the example that they did on the StereoSeq data. So they factorized their gene expression from the mouse embryo, whole mouse embryo, into gene expression modules using factorization approach, similarly to what we talked about during cell location part of our talk. And then they tried to explain, they focused on Sonic-Hedgehog pathway, which is relevant for embryo development. And they inferred both upstream and regulators, and downstream targets of this soluble ligand. Another exciting avenue would potentially be bridging what's very popular now in single cell field, gene regulatory networks, which are models of co-expressed transcription factors with downstream gene modules. So in particular, how to bridge the spatial and single cell modalities, like how to spatially map gene regulatory networks to see how they change in space, in development or cancer. And something in this direction was already done with Visium, like low resolution, like previous chemistry in this publication from Saez Rodriguez group, where it tracked spatial transcription factor and gene regulatory networks activity in a cardiac injury model. And lastly, again, bridging spatial and single cell fields. There's such thing as trajectory inference in single cell RNA-seq analysis, which allows kind of to track the transition of the cell states in single cell data in the process of, for instance, differentiation. And that is helpful to understand what's it brings some sort of pseudo time into the analysis, so kind of where the system is drifting into. And in terms of spatial dimension of such an inference, there was a work on a tool that's called ST-Learn that uses machine learning to infer and like spatially anchor the trajectory of infer trajectory that cells undergo. In this case, it was a brain injury model, and scientists tracked the astrocyte differentiation when they were acquiring a reactive phenotype to this injury along the this cortical projection. So those astrocytes were microglia acquired reactive phenotype on the cortical side of the brain where the injury occurred, and they were able to map this sort of transition spatially.
So that's what we've have to showcase for how one can look at cell-cell interactions. We hope that you were inspired by our analyses with this model data set from 10x, and hopefully you can approach your own biological questions in a similar or additional ways. So if you have any questions, we'd be happy to try to answer. I think we see a few already in the Zoom channel.
Yes, thank you so much, Valerie and Christy. That was great. Yeah, we've got a few in the Q&A here. So there's one that says, is deconvolution required when using Visium HD?
Is deconvolution required for Visium HD? So Visium HD offers a cellular and subcellular resolution, and for Visium HD, one would actually have to perform a type of image segmentation to extract the gene, like the RNA counts, and assign it to a specific cell. And then once you have this cell type, of course, you don't need to do decomposition. But I guess because Visium HD, it's a binning of different tiles, then because you have to bin certain tiles together. I haven't worked with Visium HD, but this is just what I understand from the technology. One could do deconvolution if you're not able to assign these counts to like within a nucleus or a cell boundary.
Cool. Thank you.
Hopefully that answers your question.
There's another one here that says, what is the ligand receptor database based on? Only experimental evidence or based on predictions from mass spec data?
So I think it depends on the database. Many are curated from literature evidence. This can be from in vivo models, in vitro models. I'm not
structural data, so a whole body of experimental evidence is, I believe, because it has to be like ground truth in this case. But they're expanding in terms of their modalities as well, because, for instance, like initial like cell phone DB, for instance, there were like some metabolic interactions were incorporated and such. So but yeah, they rely on, as far as I know, on the ground truth experimental data. So.
Thank you. There's another one here that says, how can you check the accuracy of deconvoluting cells? For example, if one spot contains a few lymphocytes and a few neutrophils, how can you check if that's actually the case?
That's a very good question, right? Because of course of these inference tools, one lacks this type of ground truth. So in our case, in our projects, we actually perform orthogonal imaging-based methods to confirm that the cell types that we infer from cell to location are indeed present in an adjacent section. So that's how we approach this. But these inference tools are quite powerful, I would say. And in our own experience, for example, we work with cancer samples, and we see in our experience that if we use a bulk single cell reference that contains all cancer cells from all patients, and then we map them to different Visium data of different patients, actually only the cancer cells of the same patient map to the Visium, and none of the non-patient cancer cells do. So I would just suggest that it's always good to validate your spot level Visium data with additional imaging. That's how we would approach it.
Cool, thank you. We've got another one here that says, were you initially studying lymph nodes and decided to apply these techniques, or did you choose lymph nodes for a particular reason?
So we work on lymphoma. So
yes, and short answer. We were thinking about doing different showcases with the brain data set from the lymph node from 10x versus lymph node, but then we're not neurobiologists. It was just harder to select certain pathways or so. So that's the reason why we showcased lymph node.
Nice, thank you. There's one here that says, how does the Liana Plus handle the integration of multimodal data sets in cell-cell communication analysis? And what are its advantages over traditional single-cell RNA-seq methods in terms of identifying key ligand-receptor interactions?
So in this case, Liana would provide functionalities to infer like spatial co-localization, co-expression of ligand receptors. And then it's what Christy was explaining about MISTI, that it's kind of like a multi-view framework where you could have, as we did, like one view of ligand receptors, another view of, for instance, cell type compositions or TF activities or whatever you're interested. And then you try to explain features from one view with other features from the same view or from perspective of other view, right? So it's kind of like a regression model that would try to infer what's important factors that statistically determine the particular thing. And regarding single-cell RNA-seq, is how we said in RNA-seq, like it's difficult to say which cells are actually interacting because there's like can't be like any spatial distance constraint. Yeah.